Ponizan, son of Selvadurai from group 17, and today I'm going to present about characteristics of bacteria. Okay, the first common characteristics for these bacteria is most bacterial cells, they have cell wall, and this cell wall is made up of peptidoglycan. What is peptidoglycan? Peptidoglycan is a complex of polysaccharides linked by amino acids. Okay, so most of the bacterial cells, they have cell wall like plant cells. Okay, and since the structure of cell wall is different, based on their structure of cell wall, we can divide the bacteria into two different types. Okay, first one is gram-positive bacteria. Second one is gram-negative bacteria. What are the differences between these two types of bacteria? First, gram-positive bacteria, they have thick peptidoglycan cell wall. Compared to gram-negative bacteria, they have thinner peptidoglycan cell wall. And fact about this gram-negative bacteria is they have second plasma membrane outside the cell wall. Okay, there will be a plasma membrane outside got cell wall, and another layer outside the cell wall, they have second plasma membrane. Why do they have this second plasma membrane? Is to block the antibiotic drugs, making the infections difficult to treat. Okay, that's the function of second plasma membrane for the gram-negative bacteria. Next, since there are two types of bacteria, to differentiate these types of bacteria, we will use this method called gram staining. Okay, gram staining is a procedure to distinguish types of bacteria based on their cell wall. Okay, after staining, the color of bacteria differs. Okay, after staining, after we use different substance to stain the bacteria, the color will be differing. Okay, gram-positive bacteria appears dark purple after the gram stain, whereas gram-negative bacteria appears red after the gram stain. Why does this happen? For a gram-positive bacteria, they have thick peptidoglycan cell wall. Am I right? So since they have thick peptidoglycan cell wall, it traps the crystal iodine violet. Okay, crystal violet dye. So it traps the crystal violet dye, whereas the gram-negative bacteria, they have less peptidoglycan cell wall. So it does not retain the crystal violet dye and exhibit the counter stain, which is red color. Usually it's safranin dye. Okay, that's why gram-negative bacteria appears red, gram-positive bacteria appears dark purple. Okay, the next common characteristic, bacteria can be found in three basic cell shapes. Okay, they have different cell shapes, which is basically three different shapes. Okay, first one is spirillum, which is spiral shape or helical shape. Second one is bacillus, which is rod shape, and third one is coccus, which is round or spherical. Okay, let's take a look on the example. Spirillum look like this. Okay, it's spiral shape, right? So the bacteria look like this. And next one is bacillus. Bacillus is rod shape. So you can see there it's a rod shape. And next one is coccus. Coccus is round or spherical. Okay. Next, the third characteristics for bacteria, we can characterize the bacteria by their growth arrangement. Okay, growth arrangement. First one is staff arrangement. Staff arrangement describes the bacteria cells in a cluster. Okay, cluster of cells. For an example, staphylococcus. This word staphyle means it refers to the growth of arrangement, whereas the coccus refers to the shape of the bacteria. Okay, this coccus. Coccus means the bacteria cells in round shape. Okay. Second arrangement is trapped arrangement, which refers to chains of bacterial cells. The bacterial cell will be in a chain. Okay. Example is Streptobacillus. And third one is diplo arrangement. Diplo arrangement means pairs of bacteria. Okay, the bacterial cell will be in pairs. For an example, Diplococcus. Okay. Next, why do we need to characterize this bacterial cell? What is the purpose? Because it helps to identify the species of bacterial cell. We can know what species is this bacteria. I have given you two examples. First one is Streptococcus pyogenes. Okay. And this is a gram-positive Streptococcus. Okay, gram-positive. You can see all the three characteristics in this name. Gram-positive refers to the type of cell wall. It's thick peptidoglycan cell wall. Strep means the growth arrangement. Coccus means the shape, which is round. Okay, and these bacteria causes strep throat. What is strep throat? Strep throat means a person having sore throat with fever. Okay, both condition at once. Okay. Next, second type is Neisseria gonorrhea. Okay, Neisseria gonorrhea. It's a gram-negative diplococcus. Gram-negative means thin cell wall. Diplo means refers to the growth arrangement. Coccus is round. Okay, round shape. Okay. So diplo means it's a pair of the bacterial cell. And these bacteria are responsible for the gonorrhea. Okay, it causes gonorrhea. Okay, so the references that are used is a manual textbook with 14 edition, and thank you. That's all from me. Thank you very much.
Assalamualaikum. My name is Wanuri Nabila and I will present about bacterial metabolism. So what is actually metabolism is? Metabolism is the total amount of the biochemical reactions involved in maintaining the living condition of the cells in an organism. Basically, bacterial metabolism is the process which bacteria use to stay alive. Firstly, we will go through the introduction of bacterial metabolism. There are two ways of respiration that are carried out by bacteria, which are aerobic respiration and anaerobic respiration. For aerobic respiration, bacteria require a constant supply of oxygen to carry out cellular respiration. It is a biological process in which food glucose is converted into energy in the presence of oxygen. For anaerobic respiration, there are two types which are facultative and obligate anaerobes. Facultative anaerobes are where the bacteria are able to grow either in the presence or absence of gaseous oxygen, which means they can survive in the presence of oxygen but can also survive without oxygen by fermentation or anaerobic respiration. For obligate anaerobes, bacteria are unable to survive in the presence of free oxygen, and this kind of bacteria can cause some serious illnesses such as botulism, gas gangrene, and tetanus. So we will continue to the next subtopic. For this topic, there are two types of bacteria, which is autotrophic bacteria and heterotrophic bacteria. Autotrophic bacteria are divided into two types, photoautotrophs and chemoautotrophs. The first one is photoautotrophs. Photoautotrophs bacteria use solar energy from the sun to reduce carbon dioxide to organic compounds. Photoautotroph bacteria are also divided into two, which are anoxygenic photosynthesis and oxygenic photosynthesis. For anoxygenic photosynthesis, the bacteria do not produce oxygen, while oxygenic photosynthesis bacteria will produce oxygen. Anoxygenic photosynthesis only carries photosystem 1, while oxygenic photosynthesis will carry both photosystem 1 and 2. For anoxygenic photosystem, there is one more characteristic, which is they have a unique type of chlorophyll called bacteriochlorophyll that acts as a special pair to drive the photosynthetic electron transfer in the reaction center. There are some examples of oxygenic photosynthesis bacteria, which are green sulfur and some purple bacteria. They live in anaerobic conditions, such as at the muddy bottom of marsh, and they cannot photosynthesize in the presence of oxygen and also do not emit oxygen. Besides, cyanobacteria are also one of the examples of oxygenic photosynthesis. Cyanobacteria contain chlorophyll A, and they reduce carbon dioxide to organic compounds and give off oxygen as a byproduct. The next one is chemoautotrophs. Chemoautotrophs bacteria oxidize organic compounds such as hydrogen gas, hydrogen sulfate, and ammonia to reduce carbon dioxide to an organic compound. An example of chemoautotrophs bacteria is nitrifying bacteria. The nitrifying bacteria oxidize ammonia to nitrates and nitrates to nitrates. Their metabolic abilities keep nitrogen cycling through the ecosystem. And there are also other bacteria that oxidize sulfur compounds, and they live in the deep sea, which is around 2.5 kilometers below sea level. This bacteria is important because the organic compounds produced by them used to support the growth of community of organisms at deep sea vents. Next, we will proceed with heterotrophic bacteria, which is chemoheterotrophs. The chemoheterotrophs bacteria obtain carbon and energy in the form of organic nutrients from other living organisms. So, these bacteria need to consume other organisms in order to survive. For example, parasitic bacteria feed on the tissues and fluids of their living hosts. Here are some pictures of the parasitic bacteria. Saprotroph is another name for chemoheterotrophs. This kind of bacteria will break down organic matter from dead organisms. Saprotrophs are organisms that cannot make their own food, so they obtain nutrition from dead and decaying organisms. They play a critical role in recycling matter and making inorganic molecules available for photosynthesis. Saprotrophs bacteria are also used commercially to produce chemicals such as ethyl alcohol, acetic acid, butyl alcohol, and also acetones. Besides, they are also involved in the production of butter, cheese, sauerkraut, silk, rubber, coffee, and also antibiotic. That's all for bacterial metabolism. Next, we will proceed with symbiotic relationships, which will be presented by Ayuni.
Hello everyone, so here I am today. My name is Nurul Ayuni and I'm here to present to you a symbiotic relationship. Do you guys know what a symbiotic relationship is? So, symbiotic relationship is two different species that live together in a metic way. For example, we have bacteria and archaea form a symbiotic relationship together. Type of symbiotic relationship. First, we have mutualism. Second, commensalism. And the third one is parasitism. Mutualism. Mutualism is the both species benefit from the association. For example, we have mutualistic bacteria that live in human intestine to release vitamin K and B12 to help produce blood components. Next, in stomach of cow and goats, mutualistic prokaryote digest cellulose, enabling them to feed on grass. Lastly, mutualistic bacteria in root nodules of plants help to reduce nitrogen to ammonia in nitrogen fixation process. Next, we move to commensalism. Commensalism is when only one species is benefit while the other is unaffected. Often occurs when one population modifies the environment in such a way the second population is benefit. Example, obligate anaerobes can live in human intestine if Bacterium E. coli use up the available oxygen. Next, parasitism. Parasitism is when one species benefit while harming the others. Parasitic bacteria can cause disease and therefore are called pathogens. In some cases, microbes themselves does not cause disease but what they release, which is pathological portion. For example, when a person steps on rusty nail, what will happen? When a person steps on rusty nail, Clostridium tetani can be injected into damaged tissue and produce toxin that can cause disease tetanus. Did you know that fimbriae allow pathogen to bind on a certain cell and their specificity determine which organ or cell it hooks? Many bacteria that cause severe diarrhea, Shigella dysenteria, is able to stick on intestinal wall to produce Shiga toxin that increase potential for fatality. ASPer. Some of deadliest pathogens form ASPer when face unfavorable environment condition. ASPer can survive in harsh environment. It also can survive in long period. Lastly, we move to antibiotics. Antibiotic compounds are active against bacteria and they are widely prescribed. Most antibacterial compounds fall within two classes. The first one that inhibit protein biosynthesis and the second one that inhibits cell wall biosynthesis. But you need to be careful. If use antibiotic in improper way and excessively, it can cause bacteria resistant to antibiotic. Two types of antibiotics is inhibit protein biosynthesis. It affecting bacterial ribosome because they function somewhat differently than eukaryote ribosome. Second, inhibit cell wall biosynthesis. It blocks formation of peptidoglycan, a compound necessary to protect integrity of bacterial cell wall.
Hi, Assalamualaikum. My name is Muhammad Haikal bin Asa. Today, I would like to present to you guys about cyanobacteria. What is cyanobacteria? Cyanobacteria are gram-negative bacteria with a number of unusual traits. Gram-negative bacteria is bacteria that do not retain the crystal violet stain used in the gram staining method of bacterial differentiation. Cyanobacteria also called as blue-green algae. Next is classification. Classification of cyanobacteria is eukaryotic algae. Eukaryotic cells are cells that contains a nucleus and organelles and are enclosed by plasma membrane. Now, they are classified as prokaryotics, which is unicellular organism that consists of a single prokaryotic cell. What is prokaryotic cell? Prokaryotic cells are simple cells that do not have a nucleus or other cell organelles. Next is characteristic of cyanobacteria. First, cyanobacteria cells are large, which is 1 to 15 micrometer. Next, cyanobacteria can be single, colonial, and filamentous cell. Next, cyanobacteria also lack any visible, means locomotion, which is some glide when contact with solid surface and other oscillated. Next is Cyanobacteria has special advantage because they possess heterosis. What is heterosis? Heterosis is specialized nitrogen-fixing cell formed during nitrogen starvation by some filamentous cyanobacteria. This ability is to photosynthesize and fix atmospheric nitrogen, so their nutritional are minimal. Next, we have habitat. Where is cyanobacteria can be found? Cyanobacteria are common in fresh and marine water, in soil, and moist surface, but they also found in harsh habitat such as hot spring. Effect of cyanobacteria in ecosystem. Cyanobacteria mostly affecting the ecological system in pond and lake. If phosphate drains into lake and pond, it's resulting in bloom of cyanobacteria, all called as eutrophication. The surface of the water become turbid and light cannot penetrate to lower level. When portion of cyanobacteria die off, the decomposer feeding on them use up the available oxygen, cause fishing to die from lack of oxygen. That's all for me. Thank you.
Hi everyone, today I'll be talking about the bacteria model that I've made and labeled. The first thing I'll explain is the flagellum, and it's the tail-like structure of the bacteria, and it's an organelle of locomotion, which means that it helps the bacteria move towards stimuli. Next, there's the capsule, cell wall, and plasma membrane. The capsule is the outermost layer, so it's represented by the white in this model, and it's a polysaccharide layer, and it enhances the ability of bacteria to cause diseases. The way it does this is it prevents phagocytosis by eukaryotic cells. Then we have the cell wall, which consists mainly of peptidoglycan, and it gives an overall strength to the cells. Then we have the plasma membrane, which is a selective permeability barrier that regulates the passage of substances in and out of the cell. So it controls what goes in and out of the cells, and it's composed of almost equal parts of lipids and proteins. Then we have the cytoplasm. It is where the functions for cell growth, metabolism, and replication are carried out. Then there's the ribosome, which is a micro machine for protein making. So this is where all the proteins are produced. Then there's the mesosome, which helps cells with cellular respiration. Next, we have a plasmid, which is a circular structure, and I've used a mini rubber band to represent that. And the plasmid provides bacteria with genetic advantages. An example is it gives bacteria antibiotic resistance. Then we have the nucleoid, which is a coiled structure, and I've used some string to represent that. And it controls cell activity, so you could say it's kind of like the brains of the whole bacterial cell. And lastly, we have the pilus, which is a hair-like structure involved in bacterial conjugation, attachment to surfaces, and motility.
This is how the model looks like after it's labeled.